In this presentation, we will be discussing regulatory B cells and their involvement in cancer malignancy and metastasis.
The immune system is broadly divided into two systems, distinguished by function. The innate immune system, which responds nonspecifically to pathogens, and the adaptive immune system, which has a longer response time, but tailors uniquely to the foreign invader. The adaptive immune system can be further broken down into two arms: cellular immunity, mediated predominantly by T cells, and humoral immunity, mediated by B cells.
B cells belong to the adaptive arm of the immune system and are involved in humoral immunity. Specifically, as part of the immune response, B cells secrete antibodies, pro-inflammatory and anti-inflammatory cytokines, and are competent antigen-presenting cells. There are three subsets of mature B cells: B1 B cells, B2 B cells, and marginal zone B cells. B2 B cells are the most abundant and are largely found in lymph nodes and the spleen.
While cellular and humoral immunity are often discussed as separate entities, they are actually highly coordinated systems that interact with and are interdependent upon one another. Moreover, as a greater understanding of the immune system develops, the more phenotypic subtypes of adaptive immune cells become recognized. One such novel immune cell is the regulatory B cell.
Regulatory B cells, or BREGs, are a subset of B cells and will be the focus of our presentation.
Regulatory B cells play an important role in immune homeostasis. They are one of the key modulators of the adaptive immune response. Through the release of IL-10 and other factors, regulatory B cells function to suppress inflammation and direct T cell differentiation. Regulatory B cells are largely accepted as one of the key regulatory cells in the immune system. They are able to kickstart a cascade of immunosuppressive events to halt inflammation and protect the body from its own immune system. One of the ways to truly appreciate the role of regulatory B cells in health and disease is to observe the effects when they are absent. In rheumatoid arthritis, a progressive autoimmune disease characterized by excessive inflammation and damage to the joints, patients are often deficient in regulatory B cells. Unlike healthy individuals, people with rheumatoid arthritis do not have the ability to suppress the actions of pro-inflammatory helper T cells, specifically Th17. A role that is largely accomplished by the regulatory B cells. Similar autoimmune conditions such as systemic lupus erythematosus and type 1 diabetes are also characterized by depletion of regulatory B cells.
Given their primary role of secreting factors which inhibit inflammation, regulatory B cells are of interest in relation to cancer development and progression. While it is still a very new field of investigation, several mechanisms have been identified which could help to explain how BREGs promote neoplasms. For example, it has been shown in B-cell deficient mice models that tumor growth was reduced when compared to wild-type mice. Furthermore, when BREGs were introduced to the originally deficient group, tumor growth was reinstated. It has therefore been suggested that BREGs can downregulate anti-tumor mechanisms while also promoting mechanisms which encourage tumor growth. This and other mechanisms will be discussed throughout the remainder of this presentation.
Shaw and colleagues explored the contribution of B cells to tumor growth by comparing immune responses of mice genetically lacking B cells to wild-type mice with functional B cells. A third group of B-cell deficient mice with a reconstituted B-cell population was also included. Tumor size was measured using Dal calipers 21 to 24 days after inoculation with MC38 tumors. B cells were found to possibly contribute to tumorigenesis. Tumor growth is still apparent in B-cell deficient mice, albeit at a much slower rate than control or reconstituted conditions. Analysis of tumor B-cell levels by flow cytometry depicted lower B-cell levels in inoculated B-cell deficient mice, with levels at less than half of the B-cells apparent in wild-type mice.
Additionally, immunohistochemistry analysis revealed that B-cell deficient mice had substantially higher infiltration of cytotoxic T cells compared to their immunocompetent counterparts. Cumulatively, these findings suggest that regulatory B cells within the tumor microenvironment have the ability to augment the adaptive immune response to favor tumor growth.
Immunological mechanisms of tumor suppression relies in part on the cytotoxic cell death mediated by CD8 positive T cells. This subset is primed to recognize tumor-associated antigens and can directly kill cancer cells once activated.
Therefore, disruption of cytotoxic T-cell activity via Breg-mediated T-cell death and inhibition, or by Breg-mediated conversion of cytotoxic T-cells to regulatory T-cells, will reduce the immunological stress on the tumor, allowing it to persist and eventually invade surrounding tissues.
This B-cell-mediated cancer malignancy can be primarily attributed to actions of regulatory B cells. This subset of B cells acts as immunosuppressor cells through a multifaceted mechanism. Action of regulatory B cells inhibit production of CD8 positive T cells and CD4 positive type 1 helper T cells through actions of interleukin 10. Regulatory B cells have also been found to be capable of inducing cell death in CD4 positive T cells through expression of FAS-L. Further stimulatory action of interleukin 10 on FOXP3 positive regulatory T cell production further exacerbates the loss of control over tumor growth by T cell inhibition. Production of interleukin 10 by regulatory B cells also stimulates production of interleukin 10 by effector T cells TR1, creating a positive feedback loop. Tumors may also be able to attract and convert B cells into regulatory B cells. Chemokine signaling by CXCL13 attracts naive B cells to the tumor microenvironment with expression of inhibitory molecules such as PD-L1 and cytokines tumor growth factor beta, inducing the change from B cells into regulatory B cells. Other methods may involve interactions via the CD40-CD40L pathway. This downregulation of critical tumor growth inhibitors and upregulation of other regulatory lymphocytes creates an environment permitting tumors to enter the escape phase and come out of immunological dormancy.
In addition to contributing substantially to tumor growth and proliferation, regulatory B cells have also been shown to promote metastasis in a variety of different cancers. The metastasis cascade describes the process by which cancer cells can depart from the primary tumor to other organs. The cascade involves three complex and sequential overarching processes. First and foremost, epithelial cells in the primary tumor begin to invade the surrounding stromal extracellular matrix in a process called local invasion. This process is largely dependent upon the ability of the tumor to establish and maintain an immunosuppressive environment. Once separated from the primary tumor, cancer cells can enter the lumina of the blood vessels by adhering to the endothelium in a process called intravasation. Once carcinoma cells have successfully intravasated into the lumina of the blood vessels, they can disseminate throughout the body via circulatory pathways in a process called migration. This is a relatively short-lived process and is often complete in as little as one pass through the circulation. Circulating tumor cells serve as a metastatic intermediate, which are uniquely resistant to predation from natural killer cells that survey the bloodstream. After anywhere from a few minutes to a few hours, migratory cancer cells exit the vessel, usually through a small capillary, to begin colonizing a distant organ. This is why organs with greater blood perfusion, such as the lung and liver, are especially susceptible to metastasis. Tumor-evoked regulatory B cells promote tumorigenesis and metastasis through a variety of mechanisms. Most notably, they induce the conversion of resting CD4-positive T cells to regulatory T cells within the tumor microenvironment. This suppression of immune function within the tumor microenvironment is largely attributable to Breg-mediated recruitment of anti-inflammatory cytokines such as interleukin 10 and the expression of transforming growth factor beta, which limits cytotoxic T cell activity. Notably, transforming growth factor beta signaling is responsible for the dedifferentiation and detachment of individual cells, a process that is critical for the local invasion and migration of neoplastic cells. Bregs also promote metastasis directly through the expression of STAT3, which promotes angiogenesis and thus contributes to the migration of dedifferentiated cancer cells. On a systemic level, regulatory B cells suppress the overall activity of cytotoxic and phagocytic immune cells such as T effector cells, natural killer cells, and macrophages, leading to a widespread depression of immunological action. Similarly, regulatory B cells alter cytokine production to favor anti-inflammatory conditions, allowing newly extravasated cancer cells to persist and proliferate. For instance, expression of interleukin 10 is often used as a diagnostic marker for regulatory B cell tumor invasion, and it plays an important role in inhibiting gene expression of pro-inflammatory cytokines of T cells, and the antigen-presenting capacity of macrophages, ultimately allowing for immune evasion. Cumulatively, these actions repress immunosurveillance and allow for immune evasion by migrating cancer cells. Before discussing the molecular mechanisms of breast cancer metastasis, it is first important to understand the clinical significance of these pathways. Breast carcinoma is the most common form of malignancy and second leading cause of cancer-related death among women worldwide. Breast cancer also has a propensity for metastasis, with as many as 30% of patients developing metastatic disease. Despite considerable improvements in the treatment and mortality of breast cancer, the mechanisms of breast cancer metastasis remain poorly understood. Metastasis is also responsible for the majority of breast cancer-associated deaths, where distant metastases often arise in the bone, lung, liver, and brain. Metastasis to the lung is associated with the greatest morbidity and mortality, accounting for at least 60% of breast cancer-associated deaths. The role of regulatory B cells in the metastatic progression of breast cancer prognosis is highly controversial. Some regulatory B cell phenotypes are associated with improved prognoses, while others contribute significantly to immune evasion and metastasis. In fact, as new evidence continues to emerge, the more complex the phenomenon seems to become. Knud and colleagues used in vivo murine models to describe how regulatory B cells increase the risk of pulmonary metastasis. In particular, they explored how breast cancer tumors have the ability to evoke regulatory B cells. That is, the tumor microenvironment induces the generation of a unique phenotype of regulatory B cells, called Tregs, that are critical for its ability to metastasize. By comparing T and B cell immunodeficient mice with 4T1 carcinoma, which does not have the capacity to metastasize independently, and injecting the experimental group with generated Tregs, the researchers were able to establish Treg activity as an instigating factor in the metastasis cascade. The study revealed that these modified B cells are especially adept at expressing transforming growth factor beta, leading to FOXP3 dependent conversion of CD4 positive T cells to regulatory T cells. This process, they argue, is a critical initiation point in the metastasis cascade. Notably, regulatory T cells exhibit considerable immunosuppressive functions and are key for the evasion of anti-tumor effector T cells. Since the primary article was released, their findings have been confirmed in human cancers, as regulatory T cell infiltration of breast cancer tumors have been found to be associated with an invasive phenotype and greater risk of metastases. The present figure depicts the mechanism by which breast cancer-evoked Tregs support lung metastases through the generation of regulatory T cells. In summary, healthy host B cells are recruited to the primary tumor site where they are converted to Tregs within the tumor microenvironment. The precise mechanism by which this occurs is still under investigation. From there, Tregs promote the transforming growth factor beta dependent conversion of host T cells to regulatory T cells, which aid in immunosuppression and metastatic growth. The authors argue that as long as the cancer persists, the tumor maintains the ability to generate Tregs, and thereby contribute to the metastatic cascade. Of note, cells at the primary tumor site were not able to metastasize in the absence of these immune cells, indicating their integral role in breast cancer metastasis and, potentially, as a future therapeutic target.